Interesting. Now, when I linger in front of the glass wall that separates two universes of life, I notice that there is pattern and structure to their swimming, and organization to their social lives. Even in a small tank, which is a poor substitute for the complexity of a natural habitat, fishes usually have favored areas to swim or rest in. Fishes are certainly awake, but are they aware? Being aware involves having experiences, taking notice, remembering things. An aware creature is not merely alive, she has a life. This book contains a lot of science that supports fishes being aware. But sometimes a story conveys it better than any amount of science can. Anna Negrin, a physician friend of mine from Pennsylvania, shared this account with me. It was 1989. I was snorkeling leisurely back to the sailboat anchored in the crystal clear waters off the northeast coast of Puerto Rico when a 4-foot-long grouper and I caught sight of each other. He was so close I could almost have reached out and touched him. His entire left side shimmered in the sunlight. I stopped flapping my fins and froze. We both remained immobile, suspended barely a foot under the surface, looking into each other. As I drifted with the current, his large eye moved in its socket, locked to my gaze for perhaps half a minute, which seemed an eternity. I don't remember who moved away first, but as I climbed back on the boat, let it be known that a fish and a woman had been aware of each other. Although I have looked into the eyes of whales since, I still feel this fish's presence the strongest. When I watch what fishes do, swimming through the water, chasing one another, coming to one end of an aquarium to be fed, my common sense emphatically tells me that they are conscious, feeling creatures. It goes against my deepest intuition to think otherwise. But common sense and intuition do not a science make. Let's see what the science says about sentience in fishes. The fish sentience debate. Two key players in the fish feel pain camp are the fish biologists Victoria Braithwaite at Pennsylvania State University and Lynn Sneddon at the University of Liverpool. James Rose, a professor emeritus at the University of Wyoming, denies that fishes feel pain. In 2012, Rose and 6 colleagues, each with impressive academic credentials, published a paper titled "Can Fish Really Feel Pain?" in the journal Fish and Fisheries. The crux of their argument is their belief that fishes are unconscious, and because pain is a purely conscious experience, fishes therefore cannot experience pain. The basis of their claim is what I call corticocentrism, the claim that to possess a human-like capacity for pain one must have a neocortex, the cauliflower-like portion of the brain that features ridges and infoldings. Neocortex translates from its Latin roots to mean new bark, denoting the new layer of gray matter that is thought to be the most recently evolved part of the vertebrate brain. Only the brains of mammals have it. If the neocortex is the seat of consciousness, and only mammals have one, it follows that all non-mammals lack consciousness. But there is a major snag here. Birds lack a neocortex, yet the evidence for consciousness in birds is virtually universally accepted. The cognitive feats of birds included olfacture, remembering formative locations of thousands of buried objects, categorizing objects according to combined characteristics, recognizing a neighbor's voice over successive years, using names to call one's chicks back to the nest at sunset. Inventive play, such as sliding down snowbanks or car windows, and clever mischief, such as stealing sandwiches and ice cream cones from unsuspecting tourists. So impressive are the conscious acts of birds that the nomenclature of the proverbial bird brain was overhauled in 2005 to reflect the parallel evolutionary pathway that the avian paleocortex has taken, allowing birds to function cognitively at comparable levels with mammals. Birds lay waste to the idea that a creature needs a neocortex to be aware, have experiences, and do clever things. Or feel pain. If any animal without a neocortex is nevertheless conscious, it disproves the notion that a neocortex is required for consciousness. As such, it is no basis for a claim that fishes are unconscious. There are many ways to get to a complex awareness, says the neuroscientist Lori Marino of Emory University. To suggest that fishes cannot feel pain because they don't have sufficient neuroanatomy is like arguing that balloons cannot fly because they don't have wings. Or that humans cannot swim because they don't have fins. The fish's answer to the mammalian cortex is the pallium, which is noted for its astonishing diversity and complexity. And while there is less computational power in the average fish pallium than in the average primate neocortex, it is increasingly apparent that the pallium serves functions for fishes that the neocortex does for mammals and the paleocortex for birds. We'll explore these capacities ahead, but for now, let me just mention learning, memory, individual recognition, play, tool use, cooperation, and account keeping. Returning to the hook. Let's address the situation in which a fish gets repeatedly hooked in quick succession. Stories abound of bass that are caught and released, only to turn around and be taken again the same or next day, sometimes more than once, writes fish biologist Keith A. Jones in a book for bass anglers. Some fishermen claim, understandably, that this suggests the experience of being hooked is not traumatic for the fish. Otherwise, why would they so quickly strike bait again? But hook shyness is also a term familiar to most fishermen. There are studies in which long periods of time elapsed before fishes resumed normal activity following capture by hook and line. Carps and pikes avoided bait for up to 3 years after being hooked just once. A series of tests on largemouth basses showed that they, too, quickly learned to avoid hooks and remained hook-shy for 6 months. There are also studies in which fishes resumed what appeared to be normal behavior within minutes of being subjected to invasive procedures such as surgery to implant transponders to track their movements in the wild. I fail to see how this should cast doubt on fish pain. A very hungry fish who is in pain does not cease to be hungry, so the motivation to feed may override the inhibiting effects of traumatic pain. In a 2014 interview, Culum Brown, who researches fish cognition and behavior in the Department of Biological Sciences at Macquarie University in Sydney, responded to the repeat hooking phenomenon. They need to eat. There is too much uncertainty in the world to let a meal go by. Many will strike even when they are completely full. People will often say to me, "But I keep catching the same fish." Well, yeah. If you were starving and someone kept putting a hook in your hamburger, what would you do? You keep eating hamburgers, because if you don't, you starve to death. Pain studies in trouts. The matter of hook shyness proves little, and scientists and philosophers will likely continue to debate animal consciousness for a long time to come. For probing fish sentience, we would do better to look at scientific studies on fish pain. A substantial body of research exists on the subject, of which I can provide only a small sample in a book of this scope. Among the most meticulous experiments are those performed on rainbow trouts, a representative bony fish. By Braithwaite and Snedden. Their findings are summarized in Braithwaite's book, Do Fish Feel Pain? The first step in examining the capacity for pain in fishes is to see if they are equipped for it. What sorts of nervous tissue do fishes have, and does it function as we would expect in a sensate animal? To find out, trouts were diplying terminally anesthetized and their facial nerves surgically exposed. The trigeminal nerve, the largest of the cranial nerves, which is found in all vertebrates and is responsible for sensation in the face and motor functions such as biting and chewing, was examined and found to contain both A delta and C fibers. In humans and other mammals, these fibers are associated with 2 types of pain sensation. A delta fibers signal the sharp initial pain of an injury, whereas C fibers signal the duller, throbbing pain that follows. Interestingly, the researchers found that C fibers were present in a much lower proportion in trouts than has been found in other vertebrates studied. This suggests that, in trouts at least, persistent pain following initial injury could be less severe. But the proportion skew may mean little, for, as Lynn Snedden has pointed out, trout A delta fibers act in the same way as mammalian C fibers, reacting to a variety of noxious stimuli. Next, the research team wanted to find out whether noxious stimuli delivered to the trout's skin would activate the trigeminal nerve. This was done by stimulating the trigeminal ganglion, a region where the 3 sensory branches of the trigeminal nerve converge. Microelectrodes were guided into individual nerve cell bodies in the ganglion, then 3 kinds of stimuli were applied to receptor areas on the head and face: touch, heat, and chemical. All 3 generated rapid bursts of activity in the trigeminal nerve as registered by electrical signals in the electrodes. Some nerve receptors responded to all 3 stimulus types, others to 1 or 2. This provided an important clue to the scientists that trouts are equipped to respond to different types of potentially painful events: mechanical injury, burning, and chemical damage. Being equipped to experience pain is a solid foundation for the conclusion that an organism is sentient, but it is not the final word. Even in the face of the evidence accumulated so far, it could still be that the neurons, ganglions, and brains of fishes can only register a negative stimulus in a reflexive way, without any actual sensation of pain. In the next phase of the experiments, trouts were subjected to one of 4 treatments. After being netted, then briefly anesthetized, they were either injected in the mouth with bee venom, injected with vinegar, injected with a neutral saline solution, or similarly handled but not injected. Manipulations 3 and 4 allowed the researchers to cancel out the effects of being handled and injected with a needle. The trouts were then returned to their home tank to be watched from behind a black curtain to avoid disturbing them further. The scientists measured gill beat rates, how quickly the gill covers are opened and closed, a measure known from earlier studies to be a good indicator of distress in fishes. All of the trouts were clearly distressed by the treatment they received, but not equally across treatments. In the 2 control groups, gill beats rose from an original resting rate of about 50 beats per minute to about 70 bpm. The gill beat rate rose to about 90 bpm in the bee venom and vinegar groups. All of the trouts had been trained to swim to a ring to be fed whenever a light went on, but following their respective treatments, none approached the ring, even though they had not been fed for a day. Instead, they rested on their pectoral and tail fins at the bottom of the tank. Some fishes from the bee and vinegar groups also rocked from side to side and made occasional darting movements. Some of the vinegar-treated fishes also rubbed their snouts against the tank walls or gravel, as if trying to relieve a sting or an itch. Toward the end of the first hour, control fishes' gill beats returned to normal. By comparison, gill beats of fishes from the bee venom and vinegar groups were still 70 bpm or more at 2 hours after injection, and they didn't return to normal until 3.5 hours later. In addition, at 1 hour post-injection, control fishes began to show alertness when the light came on, although they still did not approach the food ring. 1 hour and 20 minutes after injection, fishes from both control groups were approaching the food ring and taking pellets as they sank through the water. It took nearly 3 times as long before the bee venom and vinegar-treated fishes started showing interest in the food ring. The trout's negative reactions to the insults were dramatically reduced by the use of a painkiller, morphine. Morphine belongs to a family of drugs called opioids, and fishes are known to have an opioid-responsive system. Their behavior in response to it here is consistent with their experience of relief of pain by the drug. In separate experiments being conducted at about the same time, the ichthyologist Lilia Chervova at Moscow State University was documenting that nociceptors, the nervous tissue sensitive to noxious stimuli, are widely distributed across the bodies of trouts, cods, and carps. She found that the most sensitivity was located around the eyes, nostrils, tail, and the pectoral and dorsal fins, parts of the body that, like our faces and hands, do most of the sensing and manipulation of objects. Chervova also found that the drug tramadol suppressed sensitivity to electric shocks in a dose-dependent manner: more drug, faster pain relief. The experiments by Braithwaite, Sneddon, and Chervova are strongly suggestive that fishes are feeling pain and not merely responding reflexively to a negative stimulus. But there was still another test worth trying, one that would involve a change in complex behavior requiring higher-order cognitive processes. Recognizing and focusing attention on an unfamiliar object seemed just the ticket, and that is what Sneddon, Braithwaite, and Michael Gentle decided to focus on. Like most fishes, trouts recognize and actively avoid objects that have been introduced into their environment. Knowing this, the researchers built a tower of red Lego blocks and put it into the fishes' home tanks. When they returned the control fishes to their home tanks after they had been handled and injected with saline into their lips, these fishes actively avoided the tower, whereas fishes injected with vinegar regularly wandered near the tower. The vinegar appeared to impair trouts' ability to perform a higher-order cognitive behavior, awareness, and avoidance of a novel object. The research team conjectured that the pain of vinegar so distracted the afflicted trouts that they were unable to perform normal survival behaviors. In an attempt to further verify this distraction hypothesis, fishes in both treatment groups were injected with morphine following the injection of saline or vinegar. This time, fishes in both treatment groups, saline then morphine or vinegar then morphine, avoided the Lego tower. Other studies of fish sentience. The experiments I've summarized are not the final word on the matter of fish pain. There are other angles to assessing how fishes respond to what we regard as painful. One of the expectations of a consciously experienced pain as opposed to an unconscious, reflexive reaction to nasty stimuli is a variable or nuanced response. One way to test for this is to vary the intensity of the stimulus. For example, paradise fishes responded to low-level electric shocks by swimming about more actively, as if trying to find an escape route. In contrast, higher-intensity shocks led to retreat from the shock source and defensive behaviors. A different approach is to vary the behavioral state of a fish at the time of the stimulus. In a study using 132 zebrafishes, responses to an injection of acetic acid into the tail varied according to whether or not the fishes were frightened before the injection. When injected inly, zebrafishes swam erratically and beat their tails in a peculiar manner that did not produce propulsion. However, when they were pre-exposed to the alarm pheromone of another zebrafish, they behaved as zebrafishes normally do when confronted with something new or scary. They either froze in one place or swam near the bottom. They did not swim erratically or beat their tails. The difference suggests that the fish's fear suppressed or overrode their pain, a phenomenon well known in humans and other mammals. It is an adaptive response because fleeing a dangerous situation that could end in death takes priority over stopping to tend to a wound. Lynn Sneddon used what I consider to be a most convincing way to examine pain in zebrafishes. She asked if they were willing to pay a cost to get pain relief. Like most captive animals, fishes like stimulation. For instance, zebrafishes prefer to swim in an enriched chamber with vegetation and objects to explore rather than in a barren chamber in the same tank. When Sneddon injected zebrafishes with acetic acid, this preference didn't change, nor did it change for other zebrafishes injected with saline water. However, if a painkiller was dissolved in the barren, unpreferred chamber of the tank, the fishes injected with the acid chose to swim in the unfavorable, barren chamber. The saline-injected fishes remained in the enriched side of the tank. Thus, zebrafishes will pay a cost in return for gaining some relief from their pain. When Jannik Nordgren from the Norwegian School of Veterinary Science and Joseph Garner, now at Stanford University, presented a different method for evaluating pain in goldfishes, it yielded a surprising result. They attached small foil heaters to 16 goldfishes and slowly increased the temperature. Half of the goldfishes were injected with morphine, the others received saline. The authors believed that if goldfishes feel the pain of heat, then the morphine-treated fishes would be able to withstand higher temperatures before reacting to it. Not so. Both groups of fishes showed an appropriate pain response. They began to wriggle, and it happened at about the same temperature. However, checking on the goldfishes 30 minutes or more after they had been returned to their home tanks, the researchers noticed that those from each group were exhibiting different behaviors. Morphine-treated fishes swam about as they normally would, whereas the saline-treated ones showed more escape responses, including so-called sea starts, swimming, and tail flicking. Garner and Nordgren's study is evidence that a fish can feel both initial, sharp pain and the lasting pain that follows. The response can be likened to our reaction to putting our hand on a hot stove. First, we have an immediate, reflexive response. We involuntarily jerk our hand away from the heat without pausing to think about it. It is only a second or so later that we feel the true brunt of the pain. Then we may endure hours or days of discomfort while our bodies protect the offended limb and remind us not to do it again. This result suggests to me that goldfishes might have more of those C fibers, the ones associated with lasting, throbbing pain, that trouts were found to have in short supply. Towards scientific consensus. The weight of evidence for fish pain is strong enough today that it has the support of venerable institutions, among them, the American Veterinary Medical Association, whose 2013 guidelines for the euthanasia of animals state, Suggestions that finfish responses to pain merely represent simple reflexes have been refuted by studies demonstrating forebrain and midbrain electrical activity in response to stimulation and differing with type of nociceptor stimulation. Learning and memory consolidation in trials where finfish are taught to avoid noxious stimuli have moved the issue of finfish cognition and sentience forward to the point where the preponderance of accumulated evidence supports the position that finfish should be accorded the same considerations as terrestrial vertebrates in regard to relief from pain. In 2012, an August group of scientists met at Cambridge University to discuss the current scientific understanding of animal consciousness. After a day of discussion, a declaration on consciousness was drafted and signed. Among its conclusions: neural circuits supporting behavioral electrophysiological states of attentiveness, sleep and decision-making appear to have arisen in evolution as early as the invertebrate radiation, being evident in insects and cephalopod mollusks. Translation: consciousness needn't require having a backbone. Furthermore, the neural substrates of emotions do not appear to be confined to cortical structures. In fact, subcortical neural networks aroused during affective states in humans are also critically important for generating emotional behaviors in animals. Translation: emotions also derive from parts of the brain outside the cortex. And the absence of a neocortex does not appear to preclude an organism from experiencing affective states. Translation: you don't need a big, convoluted human-like brain to feel excited about food or scared of predators.